What got you into it? Was it just happened to be something you just fell upon, or?
So I am the child of a chemist. My dad was well, he's still alive, but my dad is a chemist. He taught chemistry, organic chemistry, and physics to high school students. And so, he always did science fair projects with us. And so that left me interested in science and math from jump. Always, always. And so I went to college at Tennessee State University, and I wanted to be an OBGYN until I took genetics with Dr. Elbert Lewis Miles, who's still there. And after I took his course my sophomore year, I was like, no, I want to go to graduate school. I want to get a PhD in genetics. And so I entered the MARC program, and I was in the MARC program for my sophomore year until I graduated. And, I did internships and studied abroad. And I knew the research that I did when I was in undergrad was on molecular dyneins. And they're these motor proteins that function in the movement of things, whatever things, lots of things throughout the cell. And so dyneins and molecular dyneins and their movement and their motion is important in chromosome segregation and the establishment of the mitotic spindle, etc. And so that got me interested in human diseases. And so I knew that when I went to graduate school that I wanted to study a human disease. And so I wanted to stay in the Southeast because I liked the weather and I wanted to be in a metropolitan city. So I applied to Chapel Hill, University of North Carolina, Chapel Hill, Emory here in Atlanta, and University of Alabama, Birmingham. And there was one woman at Emory that I really, really, really wanted to work with. Her name was Stephanie Sherman, and she studied Down syndrome, and which is a human disease, and it's caused by non-disjunction. And so I wanted to work with her the most based on her research and understanding why chromosomes malsegregate or non-disjoin to cause this human disease. And so just starting from that, I first started with looking for risk factors for non-disjunction as it relates to recombination. And after that, it led me to genetic factors like genes like PRDM9 and their role in non-disjunction. So anything related to non-disjunction, particularly I wanted to move from associations, things that are associated with non-disjunction, to causation, what actually causes non-disjunction. And so this is the first gene that I've been able to look at, which having a mutation in it could actually predisposed to chromosome non-disjunction. And so that's pretty cool. To me, it's pretty cool. I like it. So
You're essentially kind of on the quest to figure out what causes non-disjunction.
yeah,